Today we're going to be talking about transformation efficiency using 50 millimoles of calcium chloride compared to the original experiment of 100 millimoles. So what exactly is transformation? Transformation is the incorporation of foreign DNA into a cell. Transformation results in the genetic alteration of the cell. In this experiment, we used E. coli cells that contained a plasmid with two genes that were used in this transformation process.
The first gene that we used in the transformation process was the AMPR gene, which encodes an enzyme called beta-lactamase, which allows cells to grow in the presence of antibiotics, and in this case was ampicillin, and it's used to select transformants on LB ampicillin plates. And the second gene that we used was the GFP gene, which is the green fluorescent gene, and this allows transformed bacterial cells to glow green under UV light, and mutant GFP allows cells to glow green under white light.
Salt concentration affects transformation efficiency. The concentration of calcium chloride affects the osmotic pressure and adjusts the required ions in transformation experiments. So the higher the concentration of calcium chloride, the higher the osmotic pressure, which is unfavorable for growth. And on the other hand, the lower the concentration of calcium chloride, it's the lower the osmotic pressure, which is also unfavorable for growth. Our hypothesis was if the salt concentration is decreased to 50 millimoles, then the transformation efficiency will decrease due to low osmotic pressure within the bacteria's membrane. And as seen on this graph, the concentration of calcium chloride solution needs to be pretty favorable in order to have transformation efficiency.
This transformation efficiency calculation was pulled from our last previous successful experiment, and we used the mass of plasmid used, the volume of suspension spread, and the dilution factor, and the number of colonies observed to calculate the transformation efficiency. And that was 5.2 times 10 to the 5th colonies per microgram per milliliter for our last experiment. And on our new experiment with the variable in play, we could not calculate a transformation efficiency for this one because our cells did not grow correctly on the plates and we did not have any growth on our ampicillin plates.
So here's our results. As seen on this table, the transformed cells and plasmid with LB, we saw a lawn, and then going down this column, we see in the 1-to-1 dilution, 614 colonies, and then going down 12, and then we didn't see any at all in the last two plates. And then with the plasmid and the variable, we saw a lawn in the first plate, and then no growth at all for the rest of them. And going on to the non-transformed cells without plasmid, we saw lawn growth for both of our plates. So unfortunately, we can't come to a conclusion based on our data. What we should have seen was colonies on our LB AMP 1-to-1 with the plasmid and variable, and on the LB AMP 1-to-1 non-transformed cells without the plasmid, there should be no growth, but on our plates we saw lawn growth.
So based on our results, we may have had a couple issues with the experiment. As you can see, going from the 1-to-1 plate to the 1-to-10 plate on the ampicillin plates, this dilution is not a tenfold dilution because we went from 612 colonies on the 1-to-1 plate to 12 colonies on the 1-to-10 plate, and this hints that maybe somewhere in the dilution series we used the wrong amounts, so the dilution was not a tenfold dilution. And then on the variable plates, we had no growth on the LB ampicillin plates with the 50 millimoles of calcium chloride. So this, although we should have had a lower transformation efficiency on these plates, we should have had some growth, and we are not seeing any growth on these. And this may have hinted that we may have killed the cells somewhere along the experimental process during heat shock or thawing. And these are our references that we used in the hypothesis.